All right, so we know that it's either DNA or protein that is the molecule of inheritance. And Avery has shown that it's probably DNA, but scientists want more evidence. So in the 1950s, Hershey and Chase do an experiment using bacteriophages. And bacteriophages are a kind of virus. And viruses often have two basic parts: an outer coat called, an outer coating called, or that's composed of protein, and inside they have some sort of genetic material, usually DNA. So they have the same components as a chromosome does. They have protein and DNA in this case. And they're called bacteriophages. Phage means to eat, and bacterio refers to the fact that um, this virus attacks bacteria. So a virus is interesting in that it's not really alive, but from a biological standpoint, it doesn't have cells and it can't reproduce on its own, but it can hijack bacteria or your cells if it's a virus that can invade humans and make copies of itself that way. So the first experiment they do is to check to see if it is protein that is being injected into bacteria to cause them to make more viruses. So what do? They grow viruses in a culture of radioactive sulfur, because sulfur is one of the components of the protein in the outer surface of the virus. Okay, so they take radioactive sulfur. Why radioactive sulfur? Because with a Geiger counter, they can see where it is later on in the experiment, right? So they grow these cells inside of that radioactive sulfur. They infect some bacteria with it. Then they grind up those bacteria. They put them in a blender. They put them in a centrifuge that spins, that separates out the contents of the cell from the liquid outside of the cell, so they can see where the radioactive sulfur is. And what they find out is that the radioactive sulfur is not inside the cell. They find that the radioactive sulfur is up here in this liquid, and that liquid represents the liquid that was outside of the cell. It does not appear that the protein was injected into the bacteria. Okay, that's still not proof it's DNA, but it's evidence that it's not protein. So the protein was not injected into the cell. Okay, next experiment. Well, they do the same thing. They grow these bacteria. They grow viruses, I should say, with some bacteria in a culture with radioactive phosphorus. And phosphorus is a component of DNA, but not a component of protein. They do the same thing. They grow a bunch of these bacteria that have been infected with the virus. They grind them down, they spin them down, they put them in a centrifuge, and they look at it. And now they see that the part of I should draw the arrow here, the part of that test tube now that represents the more dense inner contents of the cell is radioactive. And what does that tell us? That tells us that DNA was injected into the cell, not protein. So they're able to say, look, it's definitely DNA that's been injected, not protein. And so they more or less provided irrefutable evidence that it was not protein as the molecule of inheritance, but it was DNA. Then, around the same time, really, Chargaff, who was interested in DNA because it was becoming an interesting molecule to study because it was probably the molecule of inheritance, came up with some rules, some very important rules. He knew the basic parts were A, T, C, and G, which we'll talk about in more detail, but he came up with this idea that DNA, we call these bases. The A, T, C, and G are referred to as bases because they actually have, they are a base. When you put them in liquid, they make it a little bit higher pH, right? So he found that ratios vary between species. For example, in sea urchins, the base A is found in 32.8% of the DNA. But in E. coli, it's found 24.7% of the time. So he sees that DNA is different in different species. He also notices that within a species, A and T are found in about the same proportion. So A equals T, and he finds that G equals C within a species. Okay, so just to show you this, here we have sea urchin. There are the bases at the top, A, C, G, and T. In a sea urchin, adenine is found 32.8% of the time. In a salmon, 29% of the time. But look, have a look at this. So in a sea urchin, adenine is found 32 point whatever percent of the time, and thiamine is found about the same, in about the same proportions. Now, those are not perfect proportions, and that could be for a number of reasons, one of which is how accurate our equipment is, right? But we can see that they're about accurate. And it happens over and over and over again. For example, we see in salmon, we see 29.7 and we see 29, right? In humans, we see 30.4 and 30.1. Now, you see there's a bunch of blanks on this table. Why? I want you to figure out what the percentages would be in those other examples. What would be in the blank spots? Using Chargaff's rules. I bet you can do it. For example, let's just do wheat together. There's a blank here for thiamine. We know that A tends to be the same as T and G tends to be the same as C. And added together, they'll both equal 100%, right? So thiamine is blank. A tends to match that, so I would guess that this would be roughly 28%. And you can do it for the rest of those as well. All right. So that's Chargaff's rules. He figures this out. Now, why is that important? Because it's going to help the next set of scientists figure out what the actual structure of the DNA molecule is. And that's where we'll take off in the next lecture video.